Welcome everybody. So the context that I want to use here is the thing that I like to do is metagenomics. We have essentially the idea that we are going to extract DNA from uncultured things. Like you got DNA and you don't know what is there, okay? We can talk a long time and maybe later, but you get the idea. You can have many questions, many cases, many projects when you extract DNA from an environment inside the body, in the metro, in the, I don't know, in a lake, whatever, and you want to understand what is happening. So the basic question is Nevar, Kim, Castane, and Napiers. So, and in fact, the last part is hard because identifying function is not so easy. In fact, let's start with the simple question, like what is there? What is there? Who? The first idea, and sometimes we do that, is using BLAST. Or some kind of alignment tool, and it's probably the best idea initial. Like, is this something that we have seen before? If your sequence matches 100% or 95% of a sequence that you already knew, great news. You are in good territory. You are speaking about people that you have seen before. But most of the interesting cases don't exist essentially because we don't know anything. We know 1–2% of all bacteria, and we sequence less of them. The nature is much bigger than what we know, what we have. So, how we can assign taxonomy or identity, give a name to things that we have not seen before? I always use the example of Australia. You know, the Australia separated from the other continent many years ago. So life evolved in a different, you have many animals in Australia that don't exist anywhere else. And you don't have in Australia animals you have in Eurasia. And I mean, you realize that lions can walk from Africa to everywhere. And you historically, you had lions in Europe and you had lions in Asia. Tigers you have everywhere, elephants you have everywhere, but not in Australia. Latin America was also similar, but Australia is much more extreme. So how you put names to animals that you have not seen before and they don't correspond. So you cannot do it very well. There is a limit, like you have a kangaroo and you can only say, okay, this is a mammal, but like what? Like a big dog or like a what? But by the way, you know what is the meaning of kangaroo, no? Huh? Everybody know what is the meaning of kangaroo? No?
No.
Means bilmiorum. So essentially the Europeans came and they say, what is that animal? What is the name of that animal? And the natives say, I don't know. Kangaroo. Kangaroo. So that give you an idea how hard it is to find names. Kangaroo. Bilmiorum. So okay, if one day I have a dog, I will call it Bilmiorum. Okay. It will be a nice name. Okay, so the philosophy here is that I mean, when two organisms are similar, are close, evolutionary close, let's say that you are your brother, you are similarly close, you will share a lot of things. They have, of course, different, but 99.99% of genome are going to be the same. And with time passes and your descendants go growing, they will start to diverge, but the more you diverge, the more different you have. In other words, the less you diverge, the more close you are, the less difference you have, and you end being more and more similar. So in fact, the first, historically, the first tool that people used here was GC content. You essentially say, okay, if two organisms have the same GC content, then they are probably the same or very similar. Okay, that was the initial philosophy. So, or let's say the other way, if two organisms have very different GC content, they are probably very different organisms. Okay, that is clear. If they have the GC content is very different, then the things are very different. You may still have similar GC content and still be different, but let's see. So GC content is content one letter. How can you make it more general? Well, you count two letters, pairs of letters, dimers, trimers, tetramers, mers, and mers of different length. And that is the origin of the word that we use, k-mer. A k-mer is a sequence of length k. You can think of it like an oligo, but the technical difference is this. An oligo is a piece of DNA that is physical and it's separated like a primer or something. An oligomer is a, well, it's a mer also, no? It's a oligo, by the way, oligo means few. Are we recording? Yes. Oligo means few. So, but typically an oligomer is something that you have it physically as a small fragment of DNA that you can hold your hand. K-mer here is not necessarily physical. It can be a subsequence of genome. It's kind of a mental idea of a sequence. But the basic point is that K-mers are of fixed size, and K-mers are going to be important. The kind of most of the research that I see in different parts for assembly and many things are going in K-mers. It's going to be a tool for many things, not only for this class. So the first thing that people do is to count the frequency of the K-mers, very much like we did before, the frequency of every letter, but here we are counting pairs of letters or 3 letters or 4 letters. For the example that I give you, I am using short K-mers, like 2 or 3 letters. But then I will in the real life, you use larger. So you have a sequence that we call S, the sequence of length L. We can call all the subwords of length K. For example, S, in our case, the sequence is either the full genome or a fragment like a DNA read. You extract DNA from the environment, you sequence, your read will have, let's say, 500 letters. 500 letters. And you cannot do better because it's what the technology allows you to read. So you either have something like a few hundred letters or a complete genome that is million letters. Okay. So you go through the genome and you take the first K letters and you say, okay, these are the, you count how many see this sequence. Then the second, the third. Essentially, you go through the genome and you take note of every combination of K letters that you see. In fact, I have a drawing later. And but before going, as before, we normalize. We get the relative frequency. We always work with relative frequencies because you don't want to be biased by the size of your sample. For example, if you have a DNA read, depending on lag, essentially, you can have it short or longer, depending on the quality, depending on many things that you cannot control. So you can have 100 letters or 300 letters. You don't want your numbers to depend on the technology. So if you work with relative frequencies, it will not depend on that. It will only be always a number between 0 and 1. So, you also have to keep in mind that the K-mers are going one by one, they are overlapping. So you have first from 1 to K, then from 2 to K plus 1. Let's see, from 1 to 10, then 2 to 11, 3 to 12, and so on. Okay? So they are not disjoint, they are overlapping. The first and the second have a lot in common. Okay, then this is the drawing I want to show you. This is the application. So you get the sequence and you get the pairs. This is not in the correct order, but you have AT, then TG, then GA, then AA. This should be, I took this from somewhere else, so okay, my mistake is taken from other. And then you go through all the sequence and you see, for example, AA, I count 4 times. I have it here, I have it here, I have it here, I have it here, 4 times. Then AC, 3 times. You have AC here, then you have AC here, and AC here. Okay? So you essentially count how many times you see pairs of letters. Okay? Here we're saying pairs because we are given an example with k equal 2. In real life, you will use 10 or 20 or something larger. Not too big, but not too small. Okay? And then you get that frequency, you take the total, and you divide and you get the percentage that they call probability here because take the course. But yeah, this is the relative frequency. Okay. So to make this, you have to make a table. So first question, what is the size of that table? That is a technical question because essentially it's how much you can count, how you count them. To count them, you kind of make the vector like we do in the R. You have to say, okay, make an empty vector of a given size. What is the size of that vector? What do you think?
The size depends on the our sequence length of our the sequence length, no? And the
no.
k value.
Depend only on the k value, because some of the element may be 0. For example, GC is not in the sequence, but the vector will always have the same size. Because you have the number of k-mers is given by the letter that you have. You have 4 combination for the first letter, 4 combination for the second. So you have 16 for 2 letters. Some of them may not occur, may not be found in the genome, but you still have them in the vector and you count them like 0. Okay? You follow me? Everybody follow me? Okay, move your head.
If the k was 3,
will be 4 to 3.
Okay, I see now.
There are 4 possibilities. So, okay. So the point is this: 4 to 3 is 2 to power of 6, which is like 32. No, yeah, no, 64. So it is a number that grows very quickly. Then you have to keep in mind that these are integer numbers. And integer numbers, let's say that you can use 4 bytes, maybe 8 bytes in the computer. So the question is, when you run out of memory, well, you can do the exercise, but I tell you in practice, when I did this, I can count k equal 10, I can count in my computer. K equal 11, okay, I can. K equal 14 took a week. Instead of an hour, took a week. K equal 15, I cannot do. I don't have enough RAM in my computer to do it, at least with the program I'm using. So many tools used in metagenomics, and Ramal and Ahmed know them, like Kraken. They also do these things. And most of the tools that people are using in this are starting with this idea that you will represent the genome not by the sequence itself, but just for this vector. And that is the crazy idea. Instead of representing the genome by 3 million letters of 30 million letters, you represent by a vector telling you the frequency of each combination of letters. So that is the key point here. We lost idea of the context, meaning that you realize that if you take the genome and you switch the order, you can change the order of the letters, and this is not going to change. You can change the order of the letters, I mean, in big blocks, of course, I mean, bigger than the K size. If you take the first half and the second half and you change the order, the genome will be different, but the vector will be the same. So we lost view of the context, but we gain a view that is easy to make it general. So for example, you can have a genome reorganization and still be able to recognize it. Now, the second point is this. In at least in metagenomics, when you sequence something, you get one of the strands randomly. So you don't know which strand you are seeing. You have both strands and you are seeing one of them. In other words, if I see ATG, I should also think that I saw CAT. Every time I see a sequence, I should also count that I saw the opposite, the reverse complement. Okay? Because it is the sequencer gives you one strand, but you are seeing both strands. So this A becomes T, this T becomes A, and this G becomes C. Okay. So in fact, essentially we need half of the vector. Essentially, we need half of the vector because for every sequence there is a paired one that will count as the same. So every k-mer is paired with another k-mer, so only the first half count. So there are some details that you can try later. I essentially say that you need 4 to k half. So if you have, let's say, 6, you need 4 to the power of 3 because you have pairs. Now there is a point that that was my first answer, but when I look further, I found that when k is even, you have some k-mers that don't have a pair. For example, this one, AATT, if you root the reverse complement, it itself. So it is not strictly true that each k-mer is paired with another. Most of them are, but there are some cases that are paired with themselves, so you cannot discount them. So the formula that I found, and I will not go in detail, we can try later if you are very curious, is like if k is even, then the number of k-mer is 4k plus 2 plus 4. And if it's odd, then in bottom line, if you have to remember, is like since the sequences are valid in both directions, you only need half. So this is the formula, 4 to the power of k plus 2. There is a small correction, but for the big idea, you can forget about it. Okay? So you can, of course, I will not do it now, but you can put in Excel k 1, 2, 3, 4 and see, well, maybe we can do it. Do we have the Excel still here? So what we say here, k, and then n, or whatever. So let's say 1, 2, 3. Okay, I'm silly. This is automatic, no? 1, 2, 3, until something interesting. So this is 4 raised to this number divided by 2. Autofill, yes. I am fine or not? Interesting. No. Okay, then you need some memory for counting these things. So you say this is let's say four bytes for each one of these. Yes. So how much memory you need? This is okay. Let's divide by 1000. Okay, let's put decimal here. So how much memory you need? Here, number. Okay, no decimal. So here you have 1 kilo, and here you have 1 mega. Okay, this thing should work. Okay, fine. How much memory you need? How much? Okay, back to the class. So when k equal 1, when this is a very extreme case, you have the point that you have four possible k-mers, A, C, G, or T. It's a one-letter thing, no? But since A pairs with T and C pairs with G, you only have two values. I mean, if I see an A, in the opposite strand, there is a T. If I see a G, in the opposite strand, there is a C. So the two values that you really can count, the two values that we got here in this thing, this number 2, are this one. Let's close this one and let's close this one. Are this. Then frequency, this one is the size of the k-mer, of A plus the frequency of T. They go together because A and T are essentially the same thing. And then the frequency of G and C. Those are the two numbers that you can count. Okay? Any question? No? Then, there is another point that since these are relative frequencies, you know that the total has to be 1. Therefore, if you know all except the last one, you can predict what should be the last one. You have one less degree of freedom. In other words, you know that the sum of A, T, G, and C is going to be 1. So at the end, you have only one independent variable. When you count k-mers of size 1, you only have one possibility, and that is the GC content. So that is what I say that counting k-mers is an idea of make it general, the philosophy of GC content. GC content, essentially you count the number of G's or C's because both are equivalent. And if you count GC, you are essentially counting AT. If you have 60% GC, then you have 40% AT. That is the balance of the thing. No? And then if you want to generalize, one way of generalize GC is to count not only one letter, but count pairs of letters or three of letters. So that is the philosophy. Counting is kind of making a larger vision of GC content. So the question here is this. The question that we want to solve is this. We have a read R to fix idea, let's say 500 letters to fix ideas. Depends on the technology, of course, but let's say 500 letters. And we want to identify what is the origin, in the sense that what is the taxonomy that better matches it. So we have a set of different taxonomies. What I mean is this. Ideally, you would like to associate to one species. So you have, I don't know, 1,000 different species or 10,000 different species, and you have the genome of the 10,000 different species. Sometimes you will not succeed with that, and you will have to look not at the species level, but at the genus level. So instead of 10,000, you have, I don't know, 500 species. Sometimes it will not be enough and you have to go higher taxonomically. So depending on how good information you have or how different, I mean, if you got something that is very, very new, you cannot classify it at the species level, but you can classify it at a higher. Like kangaroo, you can say it as a mammal, but you cannot say many other things. So essentially, the philosophy here is that you want to calculate the probability of coming from a given taxonomy, given that you know the information. The only information that you have is the read, the letters in the DNA read. So for each taxonomical unit, you want to calculate what is the probability that I got this read from this organism. What is the probability that I got R from T? And if you find what is the highest probability, and you assign to say, okay, this is the taxonomy with highest probability, therefore I assign the taxonomy of R to T. The probabilities is, of course, a long question, so we have a similar idea, calculate and score, same philosophy as before, we calculate the score. And you will see the score will probably not surprise you. So the strategy I'm going to show you is called the naive Bayes classifier, and we can talk longer one day, but not today. Just let me tell you that this is the same tool that is used by your email system to classify your email, like what is spam, what is not spam. They're using the same philosophy, the same tool. It's one of the most common tools for automatic classification of things. This is machine learning in the basic idea of machine learning, okay? And the idea, like all machine learning, you have two steps. First, you have a training. When you show examples, you have a set of genomes where you know the taxonomy, and you show them to the system. In this case, we count k-mers in the genomes for which we know the taxonomy. So we have to remember the taxonomy of every organism and this vector of k-mers, okay, for each organism. So you have organism, taxonomy, and the list of k-mers, okay, the frequency. That is the training. You make a big database for every organism. You get, what is the name, a vector, and you get the taxonomy. And in the second part, like in all this classifier, you apply this training set to a new object. This is the R that I say. So you get a read, and you apply the classifier, you get a score for every taxonomy given the read that you got. What is the score for the taxonomy? So you're not giving a score for the read, you are giving a score for the taxonomy. The read is your input, and your output is the taxonomy. And the taxa with the highest score is that you say, okay, this is the taxonomy. So you should not be very much surprised that the score is the log of the frequency. Okay? So when we train, here you notice that I put a k in parentheses to represent that you have different frequencies depending on the k-mer size. So you count how many times you see the k-mer W in a given taxonomy. That is easy to do. You take the genome and you count, like the picture that I showed you before. Okay? Then, when you want to calculate the score for the taxonomy given the read, you take all the k-mers in the read, you look in this table, you take the log, you add everything, and you get the score. There is an alternative way to write it like this one, and I will let you think about it later. Okay? Any question here? This is the hard part of the... What you should see in common between this class and the previous one is that we are counting frequencies and calculating logarithm, and then doing something with that logarithm, adding or subtracting or something. Adding, we add them. So you will see many, many methods that are based on sum of logarithm. For example, the PAM matrices and the Blossom matrices are also logarithmic, and you are adding them. And that is always the philosophy. You add logarithm. Okay? Let me show you a result. With these tools, or tools a little more complicated, but with the same philosophy, we did this work. I did it kind of 2 years ago. Oh my gosh, it got ugly now. The point is this. I was working at that time with some colleagues from Stockholm University, and they are working with archaeological DNA. And in their samples, you know, they essentially take bones or teeth from old Vikings, or they also take from Turkey. You know, Turkey is the capital of archaeology. So they extract DNA, sometimes 1,000 year old, sometimes 10,000 year old. And they, since they work with human, they use BLAST or something like that to connect the reads to the human genome and try to see what they see. And they find, for example, that in the Vikings boats, there were African people and things that are very surprising. But they found that a big percentage of their sequences are not human. A big percentage are, well, it can be contamination, but in many cases you are getting something else. So they give me that data. Maybe I can try something here. Maybe I can escape here and let me. Are you still seeing the screen? Let me make it a little narrower. Maybe that can help. Yeah. Okay. I make it smaller, but I help. Hopefully, you will get it to see the picture here. So, and then I had a tool to make this graphic. This something in R that we make this thing. So what we found, they give me all the read that they could not classify. And I applied this tool, or a tool similar to this, with a database that I got from, I essentially downloaded all the genome from NCBI, and I calculated the k-mers and I assigned this thing, and I got this classification. Some of them I could not even identify. A few of them were archaea, a few of them eukarya, this part is bacteria, but we found a lot of virus. So this was surprising. In fact, we find some virus that were not expected to be there, like Zika virus and things that are tropical. So we need to select more. But this is the kind of thing that you get. Here you see the colors correspond to the kingdom. And then you can go with the graphical version, you can click and go deeper and deeper and see how they group. Okay, maybe I didn't say this. So I have like 1 million reads. And this graphic is the summary of that 1 million read. Every read get a classification. Well, some don't get nothing. Some I say, I don't know. I don't have enough information to classify them. So the size of this box corresponds to the number of reads that are classified as virus or bacteria, or in the case of virus, you see there's some kind of dominant one. Now, the limit of the things is, of course, the limit of the knowledge that we have about virus or sequencing. We are limited by the database. This is the kind of thing that we get. So the summary is that each taxa, each organism, but not only the organism, like all the genus, all the sequences in the group are characterized by a vector of k-mer frequencies and nothing else. In this model, we only care about the frequencies of k-mer, and the rest We ignore them, for good or bad. You know that all these modular simplification, we forget that these are molecules, we forget about the 3D structure of DNA, we make a big simplification to represent this molecule with 4 letters, and now we are simplifying even more. We are not even seeing the letters, we are seeing the number of times that we see combination of letters, okay? So the question that I will ask, the question that I asked myself, and you can be asking, is what is the good K here? What is the good size of the sequence? And the answer is this: what is the good K? I essentially say, okay, how the bigger the Ks are in general, the better, but how much better? So the hypothesis that I wanted to test is this: if 2 organisms are close phylogenetically, then the profile of chimer frequency should be similar. Okay? And if they are phylogenetically different, they diverged a lot of time ago, they have different chimer distribution. Like you and your brother should have similar vectors, similar profiles, and you are, and I don't know, corn, we diverged from plants many millions of years ago, so we should have chimers that are very different, chimer frequencies that are very different. Is that true or not? Well, it depends on K, of course. So I found a database that you can take a look. Well, this is the size that they had when I look at it. This is super interesting. It's a website, and they collect all papers that can put time of divergence. So they are essentially translating taxonomy difference to time. Like, I mean, you build phylogenetical tree, but you want to have them in million years or something like that. And they find things that are non-trigger. For example, they found that what is this? Okay, this is the distance, the million years ago in the horizontal line, and these are the different what is the name? Ranks? No, what is this? Families. Some taxonomical thing. I don't know exactly if they're orders or families. And this is the line, the time that they separated from the others. So, for example, mammals at the species level, they are separated very recently. At the genus level, they separated earlier. At the family level, before, order level, and class, well, here after, no? So classes separated between 600 million years ago and 100 million years ago in different parts. And you have something like here, that you find that in some cases, families separated before genus. When you look at the genomic data, and you have this horizontal bar as the margin of error. So the ranking in prokaryotes are older than the ranking in eukaryotes, where we don't have here. Well, you take a look at the paper here on the website. Bottom line is that in this website, you can have a distance in time for different taxonomy. You can connect taxonomy with time. These are the papers, and there is an updated version. You start with an organism like human, and you see how many years ago we separated. This is human, for example. So here, the closest separation is human to chimpanzee, then hominids, then primates. For example, here we separate from the rest of primates 80 million years ago. These are the number of papers that are suggesting, supporting this information. And mammalia here, common ancestor in mammalia is 180 million years ago. And you go to eukaryota and to cellular organism. You see time, and this is the point that I took from here, time and taxonomy. So they had a lot of species, and the question is, how many of these 37 or 97,000 species have genome? And to my surprise, at least with the 2015 data, I only have 570 species with genome and in the database. Both databases are big, you know, where there are a lot of genomes and there are a lot of phylogenetic data or taxonomical data, but they have very little in common. Maybe with a little more example, we can find better. But I found 570 species. So I compare all to all, which is essentially 163,000 comparison, which was good enough to make something interesting. So I got comparing the distance. This is the phylogenetic distance of things. Some things are very common. I did the phylogenetic with the thing. In fact, it's probably not a phylogenetic, it's just a guide tree. But you see essentially three groups. You see that comparing from at this level, you got these three branches. And you will not be surprised to see that these three branches are bacteria, archaea, and eukarya. They separate. What I'm doing here is this. Maybe I'm going too fast. What I did is this. I took the genome of these 570 species. I counted k-mers. Okay? So I have a k-mer frequency for all these things. And then I compared these k-mer frequencies to see if they cluster together or not. And bacteria correspond to bacteria. The profiles of k-mers of bacteria are very different from the profiles of archaea. Bacteria are more similar to bacteria than to archaea, and they are also different from eukarya. Eukarya has much more diversity. Archaea has a good amount of diversity. I don't have many samples, that is why it's narrow. And I have a lot of information for bacteria, and they tend to be much more similar from the point of view of the frequency of k-mers. Now, the question that I had to do is how to compare two profiles. I searched in the literature and I found that the good way to calculate is this. You have the frequencies. The frequencies are a number between 0 and 1. And then you take one minus the other, and that will give you a positive or negative number. So I take the absolute value, so they are all positive. And I take, if the two sequences are equal, all these numbers are 0. If the two profiles of k-mers are very different, these numbers are big. So I have a way to calculate a distance. This is called the total variation, that is the technical name. Calculate the distance between two profiles. And this is the result. So when k is equal to 1, and I compare all pairs, okay, all pairs of organisms. When k is equal to 1, a lot of organisms are very similar. In other words, with GC content, you have a lot of organisms that have the same GC content. There are very few that have different GC content. Okay, so GC content is not good to separate organisms. A lot of confusion. With k equal to, they are more different. Essentially, you have to see here how this histogram is moving from small distances to higher distances. And then when we go to higher k-mers, when you take things that are with more sequences, you see how these columns are moving. With 11 and 12, you move to this side. So look what happened here. With Kmer 12, you have very few organisms that are different and have the same profile. The profile of Kmer frequencies for k equal 12 is very different between different organisms. They tend to be like 80% different. Let's go back. For k equal 1, taking only GC content, a lot of the cases are connected, are very similar. They're highly similar, 0% of difference. With higher case, there are nobody with the same profile. And in fact, most of them have like 80% of difference. So the last point is that you can compare this Kmer distance that I just showed you with the phylogenetic distance taken from the tree that I give you. With k equal 1, in other words, the GC content, you find this. There are many here. These are organisms that are very different phylogenetically. The phylogenetic distance is well, I don't remember the unit, but let's say 2,000 million years, let's say. I don't remember the unit now. They are very different phylogenetically, but they have the Kmer distance is small. So if you only count Kmers of size 1, you cannot separate. These things are different. This part is called confusion because all these points here have similar GC content, even if they are phylogenetically very different. So the natural question is, what happens if you got a bigger case? Well, with this is again k equal 1, and I draw a line just to have an idea. And you see that with k increasing, you have the good shape. This start to be empty, and the last one is this one. In fact, let's go to the last one, this one. Okay, well, 13 was the last that I could calculate, and you see that when the phylogenetic distance is small, sometimes the profiles are small. When the phylogenetic distance is large, then the profiles are very different. So that is a good news. It means that things that are different phylogenetically can be separated with Kmer of a good size, as much as you can. But the bad news is this: things that are very similar, that are phylogenetically very close here, sometimes, this part here, sometimes, how the drawing part here, annotate here. These are organisms that are phylogenetically very close, but they have a very different profile of Kmer. So sometimes I cannot recognize. Sometimes I have things that are very close phylogenetically, but they have a Kmer profile that is very different. So this is what I mean is this: if they are different, I can see that they are different. But if they are similar, I sometimes cannot see that they are similar. Different are different, similar, we don't know. Well, that's it. That is the method that we use for what is it? Clean everything. That is
What
a
is
method
the
that we use.
growth rate here?
Growth rate here is the shape of this line. Growth rate means the shape of this line. I mean, essentially, yeah, maybe growth rate is not the correct, but I mean, in comparison with this, with the previous slides, like I essentially fit a line. You remember that we did the log. I made a log plot, and then I find the line, and then I calculated how the thing is growing. That is like this red line is kind of the average change in Kmer distance when you increase the phylogenetic distance. And when k equal 1, the k-mer distance doesn't increase very much. But with k higher, with k 13, you follow this line. So you start with 0 because, fortunately, seeing that are phylogenetically close, they have k-mer distance also close. I'm trying to compare two distances, phylogenetic distance and the k-mer distance. And I found that when the phylogenetic distance increase, the k-mer distance increase. That is good. It means that if things are phylogenetically different, then the k-mer tool will be able to separate them. That is the good news. The bad news is that sometimes things are similar. They have small phylogenetic distance, but they have high k-mer distance. And that is something that they have to solve yet.
Do we still have those in little less k-mers, like 12, 11?
Like this one?
Yes, there are also those dots. I'm looking for those.
Yeah. Okay. That's a good question. But maybe the point is here with K. If you at the beginning, we don't have them. Well, with GC we have, but here, this part is empty. It's a good point. It's a good point. And so with K, as you say, 7, for example, with K7, things that are similar look similar. So yes, you remind me now, one of the strategies that I was thinking when I did this work was to mix long k-mers and short k-mers. So long k-mers are good for the question separating things that are very different, but short k-mers are better for the question that putting together things that are together.
That's clever.
That my intuition. I have not done it. I don't know if it works, but the soul of the idea is like some tools are better for some problems, and others are better for others. Now, Ramal was reading a paper for his final project that they were essentially, if I understood correctly, they were essentially doing this thing. So at least when I saw the presentation, I say, okay, this is the same thing. So I made the mistake of doing all this work and not publishing it because I found it was too little, too weak. But then I found someone else doing the same. So I want to see if we can mix this with the ideas of the paper that Ramal is looking, and we can try to do an updated version of this thing. So yeah, that was. Questions? The last 5 minutes before people die of sleep, hunger, lack of coffee.
Lack of coffee, yes. I'm really sleepy right now.
Yeah. On Fridays, I make big coffee. I don't drink coffee in the week, but on Friday, I drink a lot of coffee. Questions? I can imagine that many of the things are kind of overwhelming. If we are lucky, the question may come later. During the week, sometime can come. So if you get a question later, you can ask them later. The summary, if I have to say, is this. There are many tools, from the very big perspective, there are many tools that we use for bioinformatics things that are based on counting frequencies of things and calculating the logarithm and creating a score based on a logarithm. The philosophy here, the mathematical philosophy in this problem is the same as in the motif finding. And to be honest, it's the same like in BLAST. The scoring in BLAST is, again, the same philosophy. And they're based on the same idea from probability. So if one day you want to learn the real truth of life, the the real secret of the ancient magicians, the real power, we should have a probability class one day. Okay. If no more questions, you can go for lunch/coffee/nap.